Good morning. Hope you all are doing well and staying safe. My name is Ramesh Sagili. I'm an associate professor in the Department of Horticulture at Oregon State University. So my research focuses on honeybee nutrition, honeybee pollination, and honeybee health in general. So today, in the next 15 minutes, I'll try to provide some updates on Varroa research that we have been conducting for the past 2 years. This is my email address you see here. If you have any questions, you can always email me. And before I start, I just would like to acknowledge these exceptional people that have been supporting my program for the last 10 years. Some of them have left and taken other positions, but still we have several of them working in the lab. So Varroa destructor, I don't think you need any introduction. Most of the beekeepers know how devastating this pest can be. So when we talk about varroa mites, actually we have two different phases. So there is a reproductive phase where these mites are inside these cells breeding, and then we have this phoretic phase where you see this bee with mites on the body. So I know we tend not to think much about reproductive phase because we can't see these mites, but whereas the phoretic mites are visible to your naked eye, and we have techniques where we can use either an alcohol wash or a sugar shake or a sticky board to monitor these mites. But we have probably no idea on how many mites are breeding inside these cells. So here I have a animation showing you a varroa mite life cycle, and it's a refresher. I know most of you are aware of varroa mites and their reproductive biology. So here you see a mite that is dropping into the cell, and it goes into an appropriate age larva, which is about 5 to 5.5 days old, and she starts feeding on the larva and then start laying eggs once the cap is formed here by the bees. So here you see a mite that has laid eggs, and now these eggs have hatched. There is this progeny that is coming, and then there will be mating between the male, the brother, and the sisters, the females that are produced. And then eventually you'll see when the bee emerges from her cell, then this is a female mite that went in, and she's coming out with an offspring. So usually it's one mite that they are able to produce. In a worker cell, it might be two on average if it's a drone cell. So I'll not go in detail into the symptoms and the damage done by mites, but just to give you an overview for those beekeepers who are new to um, the beekeeping aspect. So PMS, parasitic mite syndrome, probably most of you heard. It's just not one symptom. There are several symptoms that you have to look at. There is the spotty brood pattern. You see there are missing cells with no brood inside. Bees will have deformed wings in some cases when the infestation is high, and there is this deformed wing virus impacting the bees. There will be larvae or pupal stages that might be visible that have been uncapped. So if you see a mix of all these symptoms, that probably is a severe mite infestation that you're seeing in your hives. So here I have a question for you. So now I talked about different phases of mites, the reproductive phase where the mites are inside these cells trying to breed, and then the bees and the mites are on the bees that are phoretic phase. So what percentage of mites are in the capped brood cells here, and what percentage is on the adult bees? Probably most of you have already pondered in the past, and so just I want you to think about this. And so I'll give you the answer for this once I get into the study that we did a year and a half ago. So I'm just giving you options. Is it 50-50, 80-20, all these different options? It could be pretty variable. Again, there is no good answer here because it changes based on the time of the year that you're looking into your hives. If there is enough brood, that means too many capped brood frames, then you may have more mites in the cells here when compared to the phoretic phase. So we'll talk about those in the next few slides. So this is the first study that I want to focus here. where we wanted to look at the Varroa population dynamics. So basically, there were two objectives. We were trying to estimate phoretic and reproductive mite population in the hives. And then we also wanted to use the opportunity to also compare these mite monitoring methods. So probably all of you are aware, we use alcohol wash or a sugar shake or a sticky board method for monitoring mites. So here we wanted to test if there, or at least compare the differences between these different methods to see if they are all similar, or we get better sense from, probably you heard alcohol wash is the best way to look for mites, but we'll see what we found. So basically we had 6 colonies that we used for this study. So between February 11th and March 22nd, these colonies received APR, which is a miticide, a synthetic miticide. Then from March 22nd until September 14th, there was no mite control. So that means they didn't receive anything for this period of time. So we just wanted to allow these colonies to grow as they can naturally, and even the mite populations were growing naturally without any control. So mite levels monitored throughout summer 2018, and then on September 14th, 2018, we went and then we killed all those hives. So basically what we did is we went early in the morning before bees were flying, and adult bees from all 6 colonies were killed using this alcohol. So I'll show you some video, at least a video and a few pictures, so that you get a sense on how this method of killing bees was taken up. So, and then all the frames with brood, they were also collected and they were stored in a freezer for evaluation for the mite. So each of those cells was opened up and looked for mite families inside those cells. So here you see we use this funnel where we shake all these bees so that they all go into this bucket full of alcohol. And that's how we kill all the bees. So probably we get 90-95% of the bees. You see some flyers are still around. So this is a video. Hope you are able to see this. So here one of the lab members is shaking the bees inside the funnel. So others are looking for brood and other parameters that we wanted to look for in those 6 hives. So here are those buckets full of dead bees in alcohol that were analyzed for mites. So basically we can get all the number of mites that are there on the bees. And then this was the most tedious or laborious part, where the students had to poke each of the cells and then see if there was a mite inside. If not, then they would not count. But if there was a mite, then we look for the offsprings as well. So it was a very meticulously done study. So it took a long time because we do a lot of other experiments. So this was not the only one for that year. So whenever students had time, we used to employ them for looking at these mites. So it took about almost a year to finish this study. So here you can see in this picture again, when they pull this brood from the cells, you can see the mites. So this is another image of a mite family that was extracted from one of those cells. You can see still a developing bee with the mite family here. So let's look at the preliminary results here. So the average number of mites in capped brood and an adult. So here on the y-axis, you see the number of mites, and here you have the phoretic phase and the mites in the brood. So this is a total of these two. So don't pay attention to this, just look for this. So I've converted for your convenience into percentage here. So these numbers. So about 41% of the mites were on the bees. That's what the phoretic phase is, right? And then about 60% of the mites were in the cells. So that's what we found in September. But again, this may not be the exact numbers you'll see at any time of the year, because this is very specific to Oregon. I know Here the participants may be from different states in the United States or even across the world. So again, you may have to, or someone has to repeat this study in your area in certain time of the year when you want to know the population dynamics of the mites in your colonies. So this is more relevant to the Pacific Northwest, or at least I would say in Oregon, where in September we are seeing this percentage, about 60% of the mites are in the brood and 41% are on the bees, which is a phoretic phase. So as I said before, we also wanted to compare all those 4 methods of mite analysis, or how beekeepers do, or in the labs, researchers look for mite levels. So here the yellow one is a field sugar shake method. The purple one is the field alcohol wash. The blue one here is alcohol wash in the lab, because in the lab we use a shaker to shake those bees that we get in the jars. So then you are able to extract more mites. There might be still some that are still attached to the bees, so they can't dislodge when you're doing with your hand shaking in the field. So you can see we get more mites extracted when we use the lab method. And then this dark bar is the sticky board, which probably I'll not talk about that at this point of time, because that's not a real comparison to these. So here you can see the field sugar shake and alcohol wash. In most of these hives that we looked at, they're pretty similar. So again, we are repeating some of this again to get a better sense, because these were just 6 colonies, but we are trying to compare these methods again in a larger study to get a bigger sample size and a more robust data set. But at least this shows, at least provides a sense that both the field sugar shake and alcohol wash look pretty much similar. So if you want to use any of those methods, it should be fine for you to get some good knowledge about your mite levels in your hives. So take home message, about 60% of the mites are in the capped brood cells during fall, especially in Oregon in September. And keep this fact in mind, again, for the new beekeepers. This is very important, because when you are using a miticide, especially something that only works for a short period of time, for example, oxalic acid, if you are using it as a vapor, and then, or maybe something, another chemical that has a lifespan of only a week or 10 days, then you may have to repeat that treatment, because you may not get a good control in that 1 week period, right? There are new bees coming every day out of those cells, and along with those new bees, you're getting new mites as well. So that's the important part I want you to think about, is anything that you use which has a shorter lifespan, the miticide, you may have to repeat those. Example, like oxalic acid. So you may have to treat your hives maybe 2, 3, 4 times, depending on where you live, with any short-term control measures. And then, as I said, you use alcohol wash or sugar shake method. Both of them look promising. So these are some current options, at least in the United States. The beekeepers have APVAR for controlling mites, which is amitraz as an active ingredient. Then there is a Mitaway Quick Strips and Formic Acid Pro. And then APGuard, which is a thymol product, HobGuard, and oxalic acid. Again, I don't have time today to go into all these different products, but probably you have knowledge of these, or you can talk to your beekeeper colleagues or the researchers in your area to find more about these products and their efficacy. So the next study that I'm going to talk is about oxalic acid vaporization study. There is a huge interest in using oxalic acid, both in the United States, Europe, and many other parts of the world. So, but then and I know many beekeepers use this as a dribble in winter time, maybe November, December, when there is no brood. But there is the beekeepers are also interested, at least I have talked to several that are interested in using oxalic acid as a vapor method when the brood is present. So we wanted to see if oxalic acid is toxic to the larva, because if you want to use it when there is brood present, we need to know if it results in brood mortality. So these were the two objectives. We looked at the brood mortality, which is basically looking at eggs and larvae after using oxalic acid as a vapor method. And then we also wanted to compare the Varroa control efficacy of both oxalic and formic acid. Again, this is not a very robust objective because we used oxalic acid only once, and it's not really realistic to compare just one oxalic acid treatment with formic, which is a proven treatment for good control of mites. So here is Carolyn Breeze, one of our research assistant. She's using this oxalic acid. You see crystals here. So this is a tool that we use. It's a battery operated tool to heat the oxalic acid. And as you know, according to the label, it's 1 gram per brood chamber. So these hives with 2 chambers will get 2 grams of oxalic acid as a vapor. So here it goes. So you put this wand in and it heats up the oxalic acid crystals there. So we also had some of our Oregon master beekeeper volunteers that wanted to assist with this experiment. So here you see, so we have this acetate sheets or transparent sheets that we can meticulously monitor the larva. So what we can do is you can put a dot over each of the cells, and you know which age larva you're looking at, and you can go back next day to see if that larva has been impacted, if the bees have removed the larva because it's having a damage due to oxalic acid. So you can meticulously look at each of those larvae that you marked. So we marked several larvae, about 50 for each age group, like 2-day-old and about 4 to 5-day-old. So you can see this dots here, you see, these are all the cells that we marked. So here are the results for that study. So we looked at the fate of eggs. So we wanted to see if the eggs are impacted by oxalic acid vaporization. So you can see percentage of eggs lost here on the y-axis and the time period here. So after 24 hours, you can see the formic acid has impacted about 35% of the eggs were gone. And then you can see in 24 hours, there is not a big difference. There is no significant difference between controls and the oxalic acid. The dark bar is the oxalic acid. After 48 hours, there was no significant difference between the controls and oxalic. Same again after a week if you look at. Then we also looked at the young larvae. So these are about 2 days old. So you can see a similar trend here. With formic, there is a significant damage. You can see these cells that were missing with these larval stages. So there were about 25% removed in this after 24-hour period in the formic, whereas you didn't see a significant difference between these controls and the oxalic acid treatments. Even after 48 hours or after a week, there was no significant difference between controls and oxalic acid. But formic, you can see there is a big damage here. About 72% of the larva, the young larva, was lost after a week. And it's a known fact. Formic is, again, I'm not saying formic is not an appropriate product. It's an excellent product for controlling mites, but you have to be very careful with the temperature range and always expect brood damage. Again, it's a balancing act here to see how much larva you want to forego for a good control of mites. So again, that's a tricky thing that you have to look for. So maybe using formic acid earlier in the season before the winter bees are being reared, so that you are not impacting the later stage colony growth. So you have to use formic acid at the right time to get a good control and still not impact your colony size. So you can see same thing with the older larva. Formic acid had a significant damage, but whereas there is no difference between oxalic and controls. So here, this graph is just to show you the efficacy of the products. Again, it's not fair to compare just one treatment of oxalic acid, one vaporization treatment with formic, because formic is known to be a very good product to control mites. You can see this orange line here, it's a formic acid. You got a very good control for mites um, in a 3-week period. So this is where the mite treatment was applied. But you see there is no significant difference between the controls and the oxalic acid. So that doesn't mean that oxalic isn't working, but one treatment of oxalic vapor probably is not enough. So we are continuing another study this year, looking at three applications of oxalic vapor 1 week apart to see if it's going to give a good control comparable to your other products like Formic or APVAR or whatever you use. So take home message from this study: oxalic acid vaporization at recommended dose 1 gram per brood chamber appears to be safe to the brood. And then formic acid treatment has some negative effect on the brood, but again, it's a very effective product in controlling varroa. So I'll summarize here with the important points that I wanted to make in varroa control, especially for the new beekeepers. As I said, varroa is the most devastating parasite that you can think of, and it has totally changed the beekeeping industry since it was introduced more than 30 years ago. So new beekeepers, please be cautious about varroa mite control. So please continuously monitor your mite levels and monitor them before and after treatments. That's very critical as well. You really want to know if your treatment is working or not. So I know sometimes beekeepers just use a treatment and assume that it's working. So I will urge you to take mite levels before you treat and after the treatment, maybe like after 2 or 3 weeks, to see if the mite treatment has worked. Because if you're not looking at that and wait for 2 months, then the damage has been done if the mite treatment isn't working. So please do monitor before and after treatments. And I have seen even a 2-week delay in applying mite treatments can cause a significant damage. So treating mites on time is critical, and I can't really emphasize more than what I'm saying now. So colonies may look robust in fall, and I see this with new beekeepers. They always say, "Oh, I had this great hive with 17, 18 frames going into winter by October, and then suddenly everything was gone. Like by December, most of the bees were dead." So again, the immune system is compromised by these mites. They're not only doing a physiological damage, but they're also transmitting several viruses. So your bee's immune system is totally compromised. And so they may look good to you when you, as a beekeeper, new beekeeper, you look at them, they may have 17 frames of bees, but their immune system is compromised. So they will die eventually by December. So just don't get, don't mistake, don't get it, don't get confused that, yeah, I saw a lot of bees in the hives and they're all fine. And probably their immune system is compromised if they had high mite infestation levels. So this is a varroa mite management guide that you can find online. It's a really nice tool if you are interested in finding a good option for you to control mites in your area. And there are a lot of great videos as well in this uh, website where you can go, honeybeehealthcoalition.org. Okay, I think that's it for today, and thank you very much. And if you have any questions, my email address is on the front slide. So please email me if you have any questions, and or else good luck and with your bees, and stay safe. Thank you.